Ja, velkommen medlemmer og ikke-medlemmer, alle som interessert i å være i dette møtet. Vi veldig glad for at det så mange som har valgt det. Mitt navn da Mai Thorseth, jeg preses, og det vil også si styreleder i det Kongelige Norske Videnskabers Selskab. Og min rolle ganske beskjeden i dag. Jeg skal introdusere litt, og så skal jeg si noen ting helt på slutten. Men i hvert fall, bare veldig kort om DKNVS, forkortelser for selskapet da. Det altså Norges eldste vitenskapelige selskap, til og med eldre enn Universitetet i Oslo. Har vært der siden 1760. Og formålet jo rett og slett å fremme og formidle vitenskapen. Så det det vi skal gjøre i dag. Og bare veldig kort om kveldens opplegg. Ja, vi ser jo titelen her, hvordan fungerer hjernen, om stedsans, tidssans og hukommelse. Og innlederne i dag, de skal snakke cirka 20 minutter hver, og vi ønsker varmt velkommen da til professor og Nobelpristager i fysiologi og medisin fra 2014, Edvard Moser, og postdoc Christy Lykken, begge to fra Kavli-instituttet. Og da blir det sånn at vi får litt foredrag på norsk, litt på engelsk, og etter de her to innleggene, da blir det vel en kort pause for å rigge om litt her. Da blir det en panelsamtale og spørsmål fra salen etter de her to innledningene, og det blir ledet av visepreses og professor i medisin, Asta Håberg. Så da nøyer jeg meg med det og sier velkommen til første innleder.
Så tusen... Dette virker ikke, gjør det? Hører dere meg? Det bra? Ok, nå virker det. Ja. Tusen takk først til DKNVS for invitasjonen, og spesielt til meg og Asta for å argumentere for at nå var det på tide med en oppdatering. Jeg synes det alltid hyggelig å snakke her. Jeg har gjort noe, det sikkert 7-8 år siden. Men det et veldig takknemlig publikum, og det det helt sikkert i dag også. Så også takk til Litteraturhuset for å stille lokalet til disposisjon. Og da vil jeg begynne med å spørre hvordan fungerer hjernen. Det naturlig et ekstremt ambisiøst og veldig bredt spørsmål. Men like fullt så det akkurat det vi ønsker å forstå. Vi prøver på Kavli-instituttet å finne ut hva de grunnleggende prinsipper for hvordan hjernen virker. Og så bruker vi da stedsans, tidssans og hukommelse som innfallsporter til å forstå det, fordi de har noen fordeler som vi straks skal komme tilbake til. Så hvis vi skal forstå hjernen, så ikke utgangspunktet helt enkelt, fordi en menneskehjerne har mellom 80 og 90 milliarder nerveceller, nevroner som vi kaller dem. Hver av de har ca. 10 000 koblinger, eller synapser som vi kaller det, til andre nerveceller. Så det et nettverk av koblinger mellom alle de 80-90 milliarder nervecellene, der hver celler har kontaktet ca. 10 000 andre. Så totalt så har vi da 10-15 nerveceller, 10-11 nerveceller og 10-15 koblinger. I tillegg da, så kommer det at disse koblingene, synapsene, ekstremt små. De under en mikrometer vanligvis. Og den kløfta, som vi kaller synapsen, den bare på 20-40 nanometer. Så jeg skal starte nå en video som illustrerer hvordan det kan se ut i hjernen, litt sånn forenklet. Her ser dere nervecellene. De sender elektriske signal, som dere ser hvitt, som går rundt i disse armene til nervecellene. Og så går det elektriske signaler ut til disse koblingspunktene, synapsene. Og når de elektriske signalene kommer dit, så blir det frigjort kjemikalier, så da krysser den synaptiske kløfta på 20-40 nanometer, og så treffer reseptorer der, så da setter i gang en elektrisk signal på den andre siden. Og det skjer da over alle de 10-15 synaptiske koblingene, samtidig på et tidsskala i størrelsesorden millisekund. Så det ikke enkelt å fange opp alt det her. Så hva kan vi gjøre da? I stedet for å gi opp, så kan vi fokusere på funksjoner, hjernefunksjoner som enkle, altså i forhold til andre ting i alle fall. Så i stedet for å begynne med det aller vanskeligste, som tenkning og språk og forestillinger og drømmer og alt sånt, så kan vi gå ned til det litt mer basale. En vei å gå sansene våre, så der det gjort mye, men det skal ikke jeg snakke om i dag. En annen vei å gå til å se på de grunnleggende intellektuelle funksjonene, og der vil jeg særlig framheve 2. Så det sted, og det tid. Det helt fundamentalt for hvordan vi opplever verden. Det kan ofte tenkes på som byggeklosser, som gir opphav til all intellektuell aktivitet. Og sånn sett så tror vi at hvis vi kan forstå grunnlaget for sted og for tid, så kan vi komme et godt stykke på vei for å skjønne intellektuell aktivitet, tenking, mer generelt. Men vi begynner altså litt nedefra, og vi begynner med funksjoner som vi alle har. Så jeg nevner i den sammenhengen også Immanuel Kant, en filosof som mange av dere er kjent med, som da foreslo at sted og tid eksempel på mentale funksjoner, mentale kategorier som man kalte det, som eksisterer forut for erfaring, a priori. Det bare der. Man kan ikke tenke seg en verden uten sted og tid. Man kan ikke oppleve verden uten sted og tid. Så uansett hva som faktisk skjer ute i verden, så er hjernen vår laget sånn at vi opplever ting i sted og tid. Derfor så er det så fundamentalt at vi har lyst til å studere det. Og så kan vi da tenke oss nå, hva hadde skjedd om vi ikke kunne oppfatte og tolke i form av sted og tid. Vel, da må vi prøve å gå litt tilbake i historien. Så la oss gå til steinalderen for eksempel, og forestille oss at her har vi en person som da gikk rundt og måtte gjøre en del forskjellige ting for å overleve. Hvis den personen ikke var i stand til å finne fram, eller ikke i stand til å huske når på dagen ting skjedde, eller når han skulle gå en plass eller en annen plass, så ville han definitivt ikke finne mat. Han ville ikke kunne rømme fra farlige rovdyr, så kanskje opptre på samme plass mange ganger, og kanskje til bestemte tider. Og definitivt ikke være i stand til å finne en partner. Sånn at det veldig sannsynlig at uten sted og tid så kan man bare ikke overleve. Og derfor, fordi det så fundamentalt, så har det her nok oppstått veldig tidlig i evolusjonen. Sannsynligvis lenge før utvikling av pattedyr, så det her viser et fylogenetisk tre for pattedyr. Vi har menneskehjernen her oppe, så har vi da gnagere og rotter her borte, og så har vi aper her, og så primater hele her, og så har vi flaggermus her, og så videre. Og alle de har mye av det samme spatiale navigasjonssystemet. Det betyr altså samme systemet for å finne fram. Og når det gjelder tid, så det litt mer komplisert, men det skal vi komme tilbake til senere. Men vi kan gå mye lenger tilbake, og det viser faktisk at noen av de mekanismene hjernen har for å for eksempel bestemme orientering, de finnes også i flue, i bananflue. Så ikke sant, det så fundamentalt at det oppstod veldig tidlig, og du trenger faktisk ikke så veldig mange nerveceller for å få det til heller. Så veldig mye har vi lært i det siste fra orienteringsstudier hos bananflue, og det fordi det så oversiktlig at vi kan snakke mer om det hvis dere vil. Men, så vi på en måte bare videreført det som allerede uttrykt i bananfluene. Så la jeg si litt mer om sted. Så når det gjelder sted, da, så må vi se på spesielt 2 områder i hjernen. Så vi har 2 områder som jeg ønsker å fremheve. Det ene hippocampus, og det andre entorhinal cortex. Så begge ligger i den mediale temporallappen. Det betyr altså i hjernebarken på utsida, men der hjernebarken folder seg inn, så det liksom litt sånn bak ørene. Da. Det langt nede, og Den har veldig mange store likheter mellom pattedyr generelt, men også mellom rotte og menneske, og rotte og mus og menneske. Så dere ser det illustrert her. Den oransje er hippocampus og entorhinal cortex i grønt. Den oransje er litt sånn diffus fordi den er egentlig på innsida, så dere må tenke at dere ser gjennom. Men det er ikke så viktig i den sammenhengen her. Så det betyr altså at med de store likhetene så kan vi nok lære ganske mye fra for eksempel å studere rotte og mus. Så, og det har man da gjort i over 100 år i eksperimentell psykologi, at man har prøvd å finne de grunnleggende prinsipper for atferd i spesielt rotte og mer nylig tid, mye mer i mus. Og her har vi da en som betydde veldig mye for vår forståelse av navigasjon, altså evnen til å finne fram. Og det er Edvard Moser, som var på University of California, Berkeley, og særlig var aktiv på 1930-, 40- og 50-tallet. Han puttet rotter i bokser og i labyrinter og så videre, og fant ut, kanskje kan vi si at det er opplagt, men det var ikke opplagt den gangen, at de kan ta snarveier og omveier etter behov. Og hva betyr det at man kan ta en snarvei? Vel, i så fall så er det ikke bare sånn at et stimulus utløser et annet, og så går det i en sånn kjede av responser. Det betyr at vi må ha et indre kart over omgivelsene for å kunne ta en snarvei. Så det var et helt fundamentalt funn, men det var ekstremt kontroversielt, og det ble det for ca. 20 år fremover, fordi man ikke hadde metodene for å gå inn i hjernen og studere hva som skjedde der. Men så kom da 1971, John O'Keefe, som vi delte Nobelprisen med. Han begynte å registrere elektrisk aktivitet i nerveceller i hippocampus, i rotte som beveger seg fritt rundt. Og det røde her er da hippocampus, det blå er entorhinal cortex i en rottehjerne. Og nå skal jeg da spille en video. Dere vil se en rotte som går rundt i en boks som har en størrelse på 1 ganger 1 meter. For anledninger så er rotter som da har implantert noen syltynne elektroder som fanger opp elektriske signal, blitt koblet til ledning, sånn at vi kan lese av de signalene på en computer. Og rotter går rundt og leter etter sjokoladebiter, og gjør det for ca. 10 minutter. Og det er et triks vi bare bruker for å få den til å besøke alle plasser i boksen. Og mens rotter gjør det da, og det hadde naturligvis ikke gjort om ikke den hadde vært veldig glad, for da leter ikke det etter mat. Så de liker det her veldig godt, men det gjør da at vi kan lytte på aktiviteten til nerveceller samtidig. Og nå skal vi lytte til en nervecelle i hippocampus. Hver gang den cellen er aktiv, altså brrr, da ser dere også en rød prikk som framkommer her på skjermen. Og det dere ser allerede nå er at den cellen i hippocampus er aktiv, elektrisk aktiv, altså 4 aksjonspotensial som vi kaller det, kun når rotta er oppe i det øvre venstre hjørnet. Ellers så er cellen helt stille. Og forskjellige celler i hippocampus er aktive på forskjellige plasser. Så det ser dere i de kartene for 10 forskjellige eksempelceller til høyre. Så rødt er høy aktivitet, blått er lav aktivitet, og du ser da at de forskjellige cellene er aktive på forskjellige plasser i den boksen sett overfra. Så det betydde da at da var det O'Keefe og en annen person som heter Lynn Nadell, som foreslo at oi, det her kan jo være Tollmanns indre kart. At de forskjellige cellene er aktive på forskjellige plasser, så kan du da, ved å lese av den samtidige aktiviteten, så kan du da faktisk finne ut hva rotta er til hver tid. Så det her var i 1975, men vi er så heldige at vi allerede fremdeles ser Lynn Nadell her på besøk på instituttet hver september, så han er her nå også, da, så det er kjempemorsomt. Så det her hadde enorm betydning, da, fordi det introduserte ideen om at hippocampus kanskje var en del av et indre kart. Men hvis det er et kart, så må det også være et kompass, og det kom da ca. 10 år senere. Så var det noen andre, vist her, som da registrert skulle inn i hippocampus, den røde strukturen her, men blingsa litt, og så havner de i det gule området her, som heter presubiculum, i stedet. Dere ser det ligger bare litt bak, litt sånn skjult. Og det de fann da, det Jim Rank fann, det var at der var cellen helt forskjellig. I stedet for å være aktiv på et bestemt sted, så var det aktiv i en bestemt retning. Så det kunne være hvor som helst i den boksen, men det var kun når rotta så i en bestemt retning. Så noen var den veien, andre var den veien, og så videre. Og til sammen så dannet det et retningskart. Så der var vi i 1980 cirka. Og da fant May-Britt og jeg ut at vi ønsket å jobbe med det her, og vi dro til Kiev, fikk opplæring, og så begynte vi da her i Trondheim å registrere fra andre celler, som gitterceller, som vi kaller det nå. Det fant vi da i 2005. Og ser dere nå igjen en rotte som går rundt i en boks her, så den går rundt i den boksen, og hver gang den cella er aktiv, så ser dere en hvit prikk nå. Den cella er nå i entorhinalkortex, altså i det blå området. Det dere nå ser, først så er det sånn aktivitetsfelt, akkurat som i den cella jeg viste i stad, den plasscella som hadde med røde prikker. Men her er det mange cluster av aktivitet, så samme celle er aktiv på mange plasser, og de plassene danner et helt merkelig, veldig regulært mønster. Så hvis dere nå ser på det andre bildet her, så ser dere i bakgrunnen, ser dere i grått, der rotta har gått over en halvtime, så den har besøkt alle mulige plasser. Og så svart, det er der cella var aktiv, og så ser dere da trekantmønsteret som da gjentar det her seg over hele miljøet. Det er det samme mønsteret som går om igjen, så det er det vi kaller et periodisk mønster. Om dere vil si at det er trekantene, eller en rombe, eller en heksagon som gjentak, så er det samme. Det kan vi diskutere etterpå, men det viktige her er at det er periodisk. Altså det gjentar seg spatialt. Og så derfor, fordi hvis man flytter seg nå i rommet, så er det forskjellige celler som er aktive. Kommer man inn i glitterfelten til en annen celle, så er det på en måte sånn at når vi flytter, så er det forskjellige celler overta, og derfor så var det mange som kalte det her en GPS for hjernen. Ikke vi, men det har noen likheter. Så er disse gittercellene også viktig å si, for det er litt bakgrunn for Christy sitt foredrag. Det er at de er organisert i subkart. Det er alltid mellom 5 og 10 forskjellige subkart, der det er forskjellige skala. Så hvis dere ser til venstre her, så har dere skala med høy oppløsning, og litt mindre oppløsning, og enda mindre, og enda mindre. Så her er avstanden mellom disse feltene sånn ca. 20-30 cm. Her kan det være over 1 meter. Og de lenger ned man hjernen går, de lenger ned i hjernen man går, de større blir skalaen. Og det går i trinnvis, som dere ser her. Det er sånn trappetrinn. Så de lenger ned man går, de mer sannsynlig er at man kommer til et nytt trinn, så hopper det opp, så blir det større skala, så går man lenger, så hopper man opp en til, og så enda en til slutt. Så det er forskjellige skala på kartet. Men nå til slutt da, så vil jeg bare si litt om hva vi har holdt på med i dag, fordi vi prøver å forstå hvordan alle disse cellene samhandler. Og det kan ikke vi forstå ved å studere enkeltceller enkeltvis, ei og ei. Vi må se på samhandlinger med nervecellene. Så langt, frem til slutten av forrige 10 år, så var det så godt som umulig. Man klarte ikke å registrere nok celler samtidig. Vi kunne måle fra ca. 10-15 celler samtidig. Nå, med ny teknologi som er kommet, så kan vi registrere tusenvis av nerveceller samtidig. Og det skyldes overgangen til å bruke databrikker for å registrere de signalene fra hjernecellene. Så i stedet for å måle på enden av en tråd eller en elektrode, der man har ett punkt der man måler, så kan man måle elektriske signal over en probe som har da, en probe som har da flere kan ha over 1000 slike registreringspunkt på samme probe. Og hvis vi da har 4 prober ved siden av, så kan vi få over 5000 slike punkt. Så det gjør at vi da kan registrere mange celler samtidig aktive. Og her ser dere da aktiviteten, det er bare et lite utvalg, 149 av dem. Så hver er da en linje her, som da er svart er aktivitet, hvitt er ikke aktivitet. Og det gjør at i stedet for å se på aktiviteten til en celle, så kan vi se på kombinasjonen av aktivitet, og det er der informasjonen ligger. Så hvis vi nå har 149 celler, da, ikke sant, og de var helt uavhengige, så kan vi da tenke oss at aktiviteten flyter rundt i et 149-dimensjonalt koordinatsystem. For det er det vi trenger, 149 akser, hvis vi skal plotte aktiviteten til 149 uavhengige celler. Og det Det introduserer et dataanalyseproblem, for det er curse of dimensionality, som man ofte kaller det. Fordi antall kombinasjoner blir astronomisk. Så hvis det er 10 forskjellige punkt på en akse på det 149-dimensjonale koordinatsystemet, så blir antall kombinasjoner 10 i 149. Antall atom i det kjente universet 10 i 80. Så da kan dere tenke at dette er beyond astronomic. Men så er vi så lykkelige at de fleste cellene er korrelert med hverandre. De følger hverandre veldig tett. Så de aller fleste kombinasjoner skjer aldri. Og da kan vi bryte den ned til hoveddimensjonene. Det finnes metoder for det, og det har vi også jobbet sammen med Matematisk institutt med. Og da har vi fått det ned til bare en sky her, der hvert punkt er da en av disse stripene av kombinert aktivitet. Hvert punkt er et sånt tidspunkt. Og så ser vi det totale skya av slike tidspunkt. Og det dere nå ser er at den skya har bestemt form. Den har formet av en torus, som vi kaller det, eller en smultring. Og den svarte stripa er hvordan aktiviteten flytter seg rundt på den skya når rotta går rundt i rommet. Så vi har altså klart vi ser da altså faktisk at det indre kartet, det her er det indre kartet, og det har en skyform, det har en torus eller smultringform, og det gjør den faktisk enda til når vi sover, så går den aktiviteten rundt på det kartet. Det slutter aldri, så det betyr en veldig viktig ting, som nå er straks ferdig. Det betyr at det kartet er uavhengig av sanseinntrykk, mest sannsynlig. Det er bare innebygd, og det er der uansett. Og naturligvis så er det kjempelurt at det kartet er periodisk, altså at det går rundt. For hva hadde skjedd hvis vi hadde et flatt kart? Da ser dere det her, ikke sant? Så da hadde rotta falt ut av verden. Så det slipper man når man kan gå rundt i 2 dimensjoner, sånn. Da må man bare forankre det kartet, men man vil aldri komme til verdens ende. Så til det her siste slide, da, det betyr altså at grunnen til at jeg tar tilbake kant, er at det her tyder veldig sterkt på at stedsanse har sterke medfødte komponenter. Det er noe vi nå direkte studerer, og svaret er ja. Så før rottebabyer åpner øynene eller ørekanalene, så er allerede kartet der, og aktiviteten går rundt på den torusen. Så det er veldig forskjellig fra det man trodde den gangen man trodde at alt kunne spores tilbake til erfaring. Så det er der vi er per i dag, og da må jeg bare si at det er mange folk som bidrar til det. Her er de som var der sånn cirka for 1 år siden, men vi er en helt fantastisk gruppe av unge forskere som jobber sammen for å finne ut hvordan hjernen fungerer. Så tusen takk for det, og la meg si til slutt da, at det er ikke bare de folka, men det er også de som betaler for det. Og der har vi sånn grupper som Kavli Foundation, vi har det europeiske forskningsrådet, Norges forskningsråd og KG Jepsen-stiftelsen, og ikke minst Kunnskapsdepartementet heter det. De gir også en ganske betydelig bevilgning til oss hele tiden. Så tusen takk for det. Og da overlater jeg ordet til Christy, som er en av de som har gjort ganske store funn i det siste. Tusen takk.
Okay, I'm Christy Lykken. It's a pleasure to speak to you here today. I'm so happy to have the honor to give this presentation. And in this part of the talk, I'm going to be telling you about episodic memory, our ability to recall the unique personal experiences from our daily lives. And these episodic memories for events and experiences contain information about what happened, when it happened, and where it happened. So episodic memory allows us to mentally revisit our experiences by recalling events as sequences that are organized over multiple timescales. So for example, when I recall a significant event, like the day of my wedding, I can remember the events that occurred in the moments as I walked down the aisle, but also those that occurred in the hours or even days, both before and after the ceremony. And for many decades, we've known that that episodic memory depends on the hippocampus, which is shown here in red. as well as several adjacent structures in the medial temporal lobe, including the entorhinal cortex, which is shown in blue. And the majority of the cortical input to the hippocampus comes directly from the entorhinal cortex. In the first part of this presentation, I'm going to be telling you about how activity in the lateral part of the entorhinal cortex, or the LEC, supports the temporal organization, or the when component of episodic memory. And in the second part of the presentation, I'll tell you about the neural codes for space in the medial entorhinal cortex and the hippocampus that are thought to support the spatial, or the where component of episodic memory. So first, how does the brain organize time? Well, we know that all experience has an element of time. We experience the world as a continuous stream of events, consisting of things that occur in a particular order and at particular places and times. The philosopher and psychologist William James referred to this as the stream of consciousness, which really nicely captures the way our thoughts, feelings, and emotions flow seamlessly from one to the next. And we can think about this a bit like beads on a string, where each bead represents a single moment in time, but the string links them together into a continuous whole. And if we want to study how the brain represents time, how should we do that? Well, one approach is that we can monitor the activity of cells in areas of the brain that might act like an internal clock, linking moments or events together into a coherent timeline. So to do this, we use the neuropixels probes that Edvard described already to monitor thousands of cells in multiple brain areas. As you can see in this image here, we can implant up to 3 probes in a single animal at the same time, yielding simultaneous recordings of thousands of cells in the lateral entorhinal cortex, as well as the medial entorhinal cortex and the hippocampus for a direct comparison. Then, using various behavioral tasks, we can study how neural activity evolves over time in each area. Now, each of the neurons that we're monitoring will become active or spike during the time period that we're listening to them. And each of those spikes is indicated over here with these colored tick marks during this 5-second segment of a much longer recording. And by looking at the activity of many neurons together, we can track how the pattern of activity in each group of cells changes over time. So if we recorded just 3 neurons at a time, we could plot the activity of the entire network with each neuron's activity along a different axis and observe how it evolves as a function of time in this 3D state space. But if we record thousands of neurons at the same time, how can we interpret neural activity that's going on in a 1,000-dimensional space? Well, as Edvard mentioned, we need to reduce the dimensionality of the data first in order to visualize it. I'll refer to a path like this one here on the right as a neural trajectory, and it essentially will tell us how neural activity evolves from moment to moment. So first, we looked at how neural activity evolves in the LEC as rats freely explored an empty arena for about 10 minutes. So the rat runs around in the box, foraging for these small pieces of chocolate that are tossed randomly into the environment. And what do we see? Here, each of the large dots represents the average activity of LEC neurons during 1 minute of this 10-minute session. And the dots are colored according to their time within the session, from light to dark. And what we see is that the neural activity in the LEC travels from the lighter colored points early in the session to the darker colored points later on. And this means that the overall activity in the LEC is continuously evolving, or moving continuously as time passes, almost like a clock. And if we look at the other areas of the brain recorded at the exact same time, we see something very different. The neural activity does not seem to drift like this over time. So at least in this context, this continuous drift within LEC neurons seems to be unique to this area. We can then ask whether this continuous drift in the activity of LEC is an inherent feature of this network, or if it only occurs as the animal is actively exploring its environment. To answer this question, we can record neural activity while the rat's sleeping. And this will tell us whether this continuous drift is a built-in feature of the network that's independent of behavior, or if it only occurs during active exploration. And what we see is that the trajectories during REM, or rapid eye movement sleep, are no different than those that we observed during awake exploration, meaning that this drift is an intrinsic feature of the network in LAC. So this means that the clock is continuously tracking the passage of time during both waking and sleep. So why is this kind of continuous drift important for our representation of time? Well, this continuous drift in LAC could help to create unique timestamps for each event. It provides a continuously changing pattern of activity that can uniquely mark each moment in time, allowing the brain to distinguish one event from another. So if we come back to our analogy of the beads on a string, we see that each bead is like a moment in time, and that the beads are distinguished from one another by the particular patterns of neural activity that are going on at that moment. But if we experience the world as a continuous stream of events, the question then becomes, how are different events distinguished from one another? In other words, how are events segmented? We know from research into the psychology of memory and perception that our conscious experience is segmented into discrete events by something called event boundaries. In our everyday lives, event boundaries can be things like entering a new room or a new place or beginning a new task. And research shows that event boundaries mark transitions within our experience, and research shows that they have a very powerful impact on memory. So we investigated how neural activity evolved in the LAC while rats performed several tasks with different types of event boundaries. So if we return to the foraging task that I showed you before, the only clear event boundary is the introduction of the animal to the arena itself. And what we see is that there's a large jump in the neural trajectory, or the path of neural activity over time, immediately after the animal is introduced to the arena, before it settles back and travels here at a steady pace. In a second task, we train the rats to run laps around this figure-8 shaped maze in order to retrieve a chocolate milk reward that was present at the top of the maze. So here, the most obvious event boundary is then the retrieval of this chocolate milk reward. And again, what we see is that there's a large jump in the neural activity in LAC immediately as the animal reaches that reward. And finally, in a third task, we see that there's a large jump in the neural trajectory the very first time that the animal encounters an object it had never seen before. And so what we see is that within the population activity, these jumps or shifts are time-locked to these event boundaries across different types of tasks, suggesting that they're a general mechanism for segmenting events. So these jumps could therefore create discrete events from an otherwise continuous flow. And coming back to our analogy, this will be like cutting the string of pearls into separate sections that each hold their own cluster of pearls. And now, we know that event memories are structured at multiple timescales. So you might remember the moment you ate breakfast this morning, as well as the fact that you ate breakfast before you got dressed and went to work. But if the activity within the LAC is continuously evolving over time, how can we represent multiple timescales of experience at the same time? To answer this question, we can return to the lap running behavior that I showed you already. And now we'll see that there are two behaviorally relevant timescales going on here. First, there's the time that it takes to complete a single lap around the figure-8 shaped track, or the trial time, which is measured in seconds. And second, there's the session time, or the time it takes the animal to complete the entire session on the maze, which is about 10 minutes. And what we see is that the neural activity in LAC repeats at the seconds-long timescale on every trial or every lap around the maze, forming a stereotyped trajectory that looks very similar trial after trial. But at the same time, we see that there's a continuous drift in the activity of LAC neurons going on at the longer timescale of the session. And what we see is that these dynamics do evolve along two independent dimensions, as I show here. So if we return to our analogy just one more time, we could see that each short loop around the coil would represent the repeating activity of LAC neurons that occurs trial after trial, sort of like the seconds hand that's moving around the clock. But because the string also wraps upwards along the coil, we can also track the continuous drift going on in LAC neurons across the longer timescale of minutes. Okay, so in this first part of the presentation, I've described 3 features of population dynamics within the LAC that have properties that are required for episodic memory. In the first, we saw that LAC activity continuously evolves over time, almost like a clock, which can create unique timestamps for each event. But we also saw that the clock can move faster at some times than at others, making a large jump or shift at each event boundary. And this can help to create discrete events from an otherwise continuous flow. And finally, we saw that the clock can track events that occur on multiple timescales simultaneously, which could help us to organize our event memories in time. Now, I've told you so far about how activity in the lateral entorhinal cortex supports the temporal organization of episodic memory. Next, I'll briefly tell you about how neural representations for space in the medial entorhinal cortex in the hippocampus support the spatial organization of episodic memory. And first, we could start by asking, well, what are the requirements of an episodic memory system? Well, first, the system must be able to store a large number of patterns, or memories in this case, meaning that it has a very large capacity. Second, the patterns for different episodes should be unique in order to reduce interference between very similar memories. And we have good reason to think that neural activity patterns in the hippocampus meet these requirements. As Edvard described already, we know that place cells in the hippocampus are active in specific regions of the environment. And if we look at the activity of an ensemble or small group of place cells, we see that their fields form a sort of map that covers the entire environment. When the animal is moved to a new room, place cells remap, meaning that they undergo drastic changes in both the location and rate at which they fire. And this is really critical. Through remapping, the hippocampus can use the same set of cells to make many independent representations. And that's exactly what we need for an episodic memory system, to store large quantities of information as independent memories. And so essentially, remapping can link the activity of place cells to episodic memory. Grid cells, which are found in the medial entorhinal cortex, provide some of the primary cortical input to the hippocampus. But in contrast to the random reorganization of place cells that I showed you on the last slide, grid cells seem to shift coherently when the animal is moved to a new room. So in this example, all 3 of these grid cells shifted their firing pattern in the same way, up and to the right, so that the relationships between each pair of grid cells is the same in both rooms. The question then actually becomes, how then can this kind of rigid transformation among grid cells produce this random reorganization or remapping of place cells? And one intriguing possibility is that grid cells that belong to different modules, meaning that they have different distance or spacing between each of their fields, might undergo unique changes between the rooms. So let's imagine this place cell that I show here in black. It receives input from grid cells in 3 different modules, and they overlap in this particular location. So this is where the place cell becomes active. If each of the modules shifts independently between the rooms, as I show here, the location at which these patterns overlap will change, and the place cell will become active or remap to a new location. And this question, what mechanism underlies the remapping of place cells, has been an open question in our field since the discovery of grid cells back in 2005. But it's been a very difficult one to answer because these different modules of grid cells are so spread out anatomically spread out along the dorsal to ventral axis of the MAC. So for many years, the recording technologies that we used were confined to recording a small location just at the tip of the recording device, which made it really difficult to sample from multiple modules at the same time. But now, with recent advances in recording technology, we can record along the entire length of the probe all at once. So we can record from grid cells, lots of grid cells, in multiple modules at the same time. And in doing so, we've been able to provide the very first evidence that grid cells or grid modules do shift independently between rooms, and that this functional independence of grid modules coincides with the remapping of place cells, and may therefore be the mechanism that underlies remapping. And we can think about this a bit like a language. So here, if we think of the spatial phase for each of the grid modules as a letter in a specific position of the word, the representation at this level will be low-dimensional. So each of the modules will be restricted to one of the 26 options available in the English language alphabet. But downstream in the hippocampus, the number of representations or words that we can generate using these letters is enormous. And each one conveys meaning. These maps convey both spatial and non-spatial information about the events and experiences that occur in each environment. Okay, so in the two projects I've told you about today, they share in the same main goal, in understanding how our representations for space and time support episodic memory. And this knowledge is critical because we know that place and time are the building blocks of episodic memories. And from famous case studies like that of patient HM, we know that episodic memory depends on the hippocampus and associated structures in the medial temporal lobe. Back in the 1950s, HM underwent a surgery that removed his hippocampus in both hemispheres in order to treat his ongoing epileptic seizures. But following the surgery, he had a profound memory impairment and was unable to form new episodic memories for the rest of his life. And his pattern of memory impairments was very famously studied in the lab for several decades until his death in 2008. And this research was fundamental for our understanding of episodic memory. We also know that Alzheimer's disease, the most common cause of dementia, typically begins as a gradual loss of episodic memory before affecting other cognitive functions like language and attention. So perhaps it makes sense then that the entorhinal cortex and the hippocampus are among the very first brain areas to deteriorate during the disease. First, fibers within the nerve cells clump together and form tangles. And then these fibrous tangles spread throughout the rest of the brain, causing cell death and an overall loss in brain tissue volume. Alzheimer's disease is currently estimated to affect more than 50 million people worldwide, and we know that age is the most important risk factor for the disease. So most cases begin after the age of 65 or so, and the risk roughly doubles every 5 years or so after that. By 2050, it's estimated that around 100 million people will be living with dementia. And this is expected to strain both healthcare systems and caregivers because the disease is so persistent, disabling, and costly. So for these reasons, it's critical that we understand how our representations of space and time support episodic memory, not only in mice and rats, but also in humans. Translational research in humans, both healthy and sick, began in 2016 with the help of the Research Council's infrastructure program and a collaboration between NTNU, St. Olavs, and Helsemeet, which enabled the acquisition of Norway's first 7 Tesla MRI. And using MRI in combination with tasks like movie watching or virtual navigation, we can further our understanding of questions like how do humans encode time and create maps of space. And in doing so, we can one day learn to detect the earliest signs of Alzheimer's disease and design strategies that can prevent or slow down cognitive decline. And we have very good reason to hope. In the 118-year history of Alzheimer's disease, the progress has never been more rapid. Recent advances in treatment, prevention, and early detection all have the potential to change our perspective on this devastating neurodegenerative disease. And with that, I'd like to say thank you again for inviting me to speak here today, and a big thank you to everyone listed over here on the right who are other members of the Moser Lab who contributed on both of the projects that I talked to you about today. None of the work would be possible without everyone in the group participating together. Thank you very much.
Thank you, Christy and Edward. There will be a short break where you can go and buy something to drink, and we will reconvene in 5 minutes. thank you for coming back in time. So we'll start with the discussion. In order for the discussion to flow more normally, we will speak in English, but you can ask the questions in any language that I can translate. And I'll first like to thank both of you. It's very pedagogical talks. I feel like it was, I really learned a lot, so thank you very much. I'll ask a couple of questions first, and then we'll take questions from the audience. So I'll start with you, Edvard. So you have basically worked on space primarily throughout your whole research career, and now you have added the time into the mix. And what's the reason for this, and how can you, is it still exciting?
So the reason for starting with space actually already in the 1990s is that at that time was one of few cognitive brain functions that were reasonably well understood, and there was the discovery of the place cells. It was one of very few examples where you actually could relate activity of single cells to something observable and easily measurable in rats. So I think that was our way into the cognitive brain. And that field then has developed enormously. So as I said, it is a window into understanding intellectual brain functions. Now, time is just as important, but that is a research field that is much less mature. It's very... one of the problems is that time is everywhere in the brain. Every cell in the brain has to deal with time in some way or the other. And there are many different ways of time. Time is something that just happens because things have to happen in a certain order. But it's also what we interpret as time, which is a highly advanced cognitive activity that maybe only humans do, to some extent at least. So it has been really difficult to understand it. But I think now is a good time because neuroscience is developing so quickly and there's so much exciting happening that we actually can understand even such a complex function as time. And then, as Christy mentioned, this is all tied together in memory. So if we want to understand memory, we also must understand time. It's not sufficient to understand space.
So Christy, we all have a sense of time, how it's passing at different speeds, and is it really like chronological time, or are we remembering order? So what are your thoughts about that?
Yeah, that's a really interesting question. So I think one important point, at least, that I didn't have time in the talk to make is that this representation of time that we describe in the LEC is certainly not linear, and it doesn't move the way that clock time moves. We can think about it as more of an episodic time, and in that way it's probably very subjective, person to person, and with even within an individual, depending on the certain experience that you're having. So you might have the experience that it seems that time is flying by or time is moving at a standstill. And I think what's really exciting about the projects that we're working is we can try to understand exactly what's going on in the population dynamics that can underlie these very sort of subjective feelings that we have. And we think that this is really important when we talk about these different shifts within or jumps within population dynamics, that when these types of things happen, especially when they're associated with an event boundary, it can really impact your recall of the memory, your memory really, of the distance of how far apart two events were in time.
Thank you. So I actually attended the talk at the Kavli Center last week, and I heard you say something about the importance of connecting function and structure in the brain. And I wonder, it's like since I'm doing MRI, it's a huge structures compared to when you're measuring this electrical activity. So what scales should we measure structure-function relationships at?
I think there is not one scale that is the right one. I think we have to understand it at all scales. So we can understand any brain function by looking at individual cells and how they calculate the combination of electrical signals and so on, and then produce an output. That's one thing, one level of understanding. Then we have to know how all these thousands of cells interact, and that's where we do most of our work. But that's also not sufficient, because if you want to relate that to global functions or parts of the brain, you can't record from all of those cells. Then sometimes it's convenient to have more global measures, like in fMRI. And also in humans, you rarely can get the individual cells, right? So then you have to take the second best, if you understand networks, which is small cubes of activity that still are quite representative of what's going on. And then at the top, you can't do all of that without studying behavior. So you have to do this while we are doing behaviors. So only when we put all of this together, we can understand the brain. And therefore, neuroscience is one of the sciences that is most interdisciplinary of all. I mean, it didn't exist when I was a student. Then it was you either did biology or medicine, or you did philosophy or chemistry, and now it's much more computer science, it's mathematics, it's physics, so it all comes together, and there's not a single person who can do that all, so we have to work together. Yeah.
So you were showing the remapping, Christy. So is that what happens when you come someplace that you can't really remember where you're supposed to go, or?
Yeah, that's a really interesting question. So I think it can really depend on if you are entering an environment that has changed and you feel disoriented or you don't remember exactly where you are. I think it probably really depends on how different that environment is. So if you imagine something like a rearrangement of your familiar grocery store, if they just changed the contents of a couple of the aisles around, you might not want all of your place cells to form a new map. You don't want a completely new map in that instance. You might want to keep some portions of the map the same while changing the locations where there have been changes within the environment so that you don't go down the wrong aisle. And we have some evidence that place cells actually do this. We would call it a partial remapping. So some portion of the map remains the same and it changes in other places. But in the other case, at least for my familiar grocery store, was completely remodeled recently. So in that case, maybe what I want to do is lay down a completely new map because the entire layout of the store has changed. And while I don't know for certain that that's the way that this works, I think we have a good intuition that that's probably true.
I think what I find fascinating is how many maps you can have. So you never run out of maps. So I don't think you'll ever get to the point that you have to recycle a map because you don't have more of them. I mean, it's endless. And that's because of the number of combinations, which is so astronomical that you can always create new combinations.
Yeah, absolutely.
But humans can be quite different when it comes to how good they are at finding their way and remembering things. Are rodents similarly good or bad at it?
Yeah, absolutely. So I think that that's interesting. We know that with selective breeding, especially of lab animals, that we're going to reduce some amount of genetic variability within the population in terms of both physical and cognitive traits. But that being said, I think I've been working with rodents for 17 years now, and I'm still surprised at the differences among animals. Some are very quick learners, some are more naturally curious or exploratory. And in any task that we as humans design, the animals tend to find their own solutions to these sort of complex problems. So I think that's a really interesting aspect of their behavior, and it keeps us as researchers on our toes.
Thank you. I think we can open for some questions. There's a microphone. Anyone who wants to ask a question?
Yes, thank you very much. There is a lot of progress going on with stem cells, reprogramming of stem cells, and you can grow brain organoids. How do you look at that as a model system to also understand functions of the brain in the future and now?
Do you want to go ahead?
Shall I go ahead? Yeah, I think here you're speaking about future to a large extent. I think there will come a day when you can reprogram stem cells and perhaps also introduce and grow new cells to replace defect cells. But I think at the moment, I think to reprogram cells, then you need to know what to reprogram to. And I think for most cells and most brain functions, we still don't know really what to reprogram to, and we don't really know the gene expression patterns of those cells. So I think it is still a little bit early. It's probably, well, I would say we're lucky that the brain is more complex than the kidney or the liver, but that's where we are. It's still early days, and I think that day will come one day, but we're not there.
Yeah, I think, and to build on that, I think that's a great answer. And I think, especially to Edvard's point, the functions that we study are so complex, these cognitive functions of our memories and our subjective sense of time, how we would reprogram stem cells to do what are actually very basic human functions that we still don't understand as neuroscientists is certainly something that could be down the line, but maybe not right on the frontier as we speak.
Yes, thank you. So I was curious, I think you touched a little bit upon it by rats and humans. So, the biggest challenges going from the rats and into humans, you talked about like the big MRI and that you can also look into the human brain. So what are the biggest differences in the terms of rats to humans and that translation? And can also, a follow-up question, can the rats also lose memory in the same sense that humans lose their memory as in Alzheimer's, for example?
Yeah, that's a great question. So I think one of the main differences, and especially for some of the research we do, is that with humans, you can ask them what their experience was like. So there is some really beautiful overlap, particularly between the project where we look at population dynamics in LEC and how they evolve over time. There's actually a rich body of evidence that comes from humans, fMRI studies and behavioral studies in humans that we can look at and we can sort of, in broad strokes, understand, okay, these are some predictions about what we think the underlying neural activity must look like if these things are true, and we're looking at them at a very broad level, say with fMRI. And so in animals, you have to be really careful thinking exactly about what behavioral task you want to do, exactly which function you're tapping into, because you can't ask the animal afterward, like, is that the strategy that you used? So there's a little bit more of being careful in that particular situation.
I guess you also asked about if animals can forget.
Oh, yeah, great. Yeah, so that's a great, you had a second part of your question. So you asked if animals can have impairments in their memory, and that is certainly the case. There's a lot of research going into studying aged animals or animals that are Alzheimer's disease models, where maybe the researchers manipulate a gene that's very strongly associated with Alzheimer's disease. And there's a lot of different behavioral tasks that you can do with these different groups of animals and see that their spatial memory behaviors are impaired. And we have some good reason to think that it does tap into what we would refer to as an episodic-like memory function, though we can't say that there's a direct correspondence between human and animal episodic memory.
Thank you for a very good presentation. I wonder about state-dependent learning. That case, for instance, if you drink alcohol while you're studying, when you have exam, you should also have some alcohol to get it through again. Is that a myth or is it real? And another question is about emotions. It seems like you are more able to get the memory if you have a high intensity of emotion, that it's better for you to memorize. And the third part is also the smell. It seems like if you have smell of something, you can remember even though it's 30 or 40 years ago. So that Is that the same? Have you looked on those aspects, state-dependent learning, emotions, and smell, how it's connected to memory?
That's a great question. So we haven't looked at that specifically, and in some way the different parts of your question all, in my mind, kind of relate to the same thing, which is when we think about this drifting population activity in the LAC, we can think of this, and in the psychology literature, we would refer to this as a temporal context signal. So it could be the case that if you're in the same situation as you were when you learned the material, that context activates the pattern of neural activity that might be very similar to the one when you studied the material. And that might make recall of the things easier. Often this can be referred to as a contiguity effect. So if I trained you on a particular list of words, and then sometime later I ask you what words did I teach you, and you recall one of them, you might be in a temporal context that's very close. It causes you to remember the very next word in the list. So I don't know exactly about how that pertains to drinking alcohol when you're studying, but who knows?
But you do point out something important, namely emotions. So I think we all remember probably what we did on September 11th in 2001. So I can sort of recall the whole day because it's a strong emotional experience, right? And then it's encoded and it sits there, it's stuck. And the same with traumatic experiences very often. The emotional load of the experience is also very important, because what happens in such situation is there's a lot of neurochemicals that has come from the brainstem and spread all over the brain, and these make the memory stick much better than in, for example, an eventless task.
Mm-hmm. Should we say something about olfaction too?
Oh yeah, olfaction. You want to, or shall I?
Oh, go ahead.
No, I mean it's true that olfaction is something that can last extremely long, because they, especially if they're unique. And there was a colleague of us who studied this phenomenon because the olfactory memories can be extreme sometimes. So this was in the UK, and he interviewed people who had visited the York Viking Museum, and then he presented them with an odor, and then he didn't say anything about the context, and then many of them could say immediately, this is for the Viking odor from the York Museum, because it was so special. So that just illustrates the importance of odors. But of course everything can trigger a memory. It doesn't have to be an odor.
Yeah, thank you for a very interesting presentation. You were talking about place here.
Oh, there. Okay. Yeah.
And direction. And I'm wondering, what does the magnetic field mean for that? Because some of us always know where north and south is, and birds, migrating birds, use the magnetic field, I've been told. So what does that, does that come into your research?
Yeah, it's a very good point. It is clearly species dependent. So even in mammals, some mammals can do this. So bats are really good at it. So they can use magnetic north to navigate and travel over long distances. So even in the absence of, for example, information from the stars or whatever, they can find their way. But and the same with birds, of course, although their brains are quite different from ours, at least somewhat different. But with humans, I think the effect is quite negligible. And I don't think I know of any evidence that it really makes much of a difference to human navigation. I don't know. Do you know anything else, Christy?
No, not so much. I think some indications that we actually get are from the recordings that we do when we can record, say, place cells and head direction cells at the same time, and we move the animals from one room to the other. I, as the experimenter, know what the relationship is in terms of the cardinal directions between those two rooms, and I might often see that a population of head direction cells rotates. So maybe north in one environment is a different north than in the second environment. So it's some indication to us that it's really not based on the magnetic sense in the animals, and instead that the process is more, again, kind of subjective, animal to animal, and that they're using some feature within the room itself to lay down the map and orient themselves within this like somewhat more local space rather than in the larger environment as a whole, because we aren't monitoring their navigation in that way.
Okay, do you have a theory or an explanation of why in Alzheimer's disease, why the shortest memory disappears first, and then this clinical experience that the last memory, the earliest memory for a human being disappears last in Alzheimer's?
Go ahead.
Yeah. Shall I?
Yeah, go ahead.
Yeah. No, so a common explanation of this phenomenon, it doesn't only apply to Alzheimer, it applies to all kinds of damage to the memory systems. So a common explanation is that over time, over years, the brain still continues to work with the memories and maybe reinterprets it, puts it in a new context, which has the advantage that you store the memories, they get more and more robust. It's much more harder to get rid of them because they're spread all over the brain. The disadvantage is, of course, that as this process goes on, we are rewriting our own history, and what comes out at the end may be quite different from what actually happened. But that's a, yeah.
Yes, when it comes to the implications for human treatments, I was reading just the other day about the dementia city in the Netherlands that you're probably aware of. Like instead of having patients with dementia in a room or in a small institution, like they have a whole part of the city where they can just go and wander and go and shop and so on, and everybody there attending are health personnel. So in terms of talking about the remapping of the place cells, I was wondering if besides the soothing effect of familiarity, this stimulation of the remapping can actually have some sort of treatment effect in order to avoid the progression of the symptoms?
Yeah, I think you're thinking about that in just the right way. I think there's a lot of these types of studies where you can, using physical exercise or even mental exercises, it's sort of like the more you use those muscles, whether they're physical muscles or your cognitive ones, the stronger the associated function can become. So I think in situations like those ones where you're allowing participants to move about in the world and use their spatial navigation abilities, they have more likelihood to show changes in hippocampal volume, increased functional and synaptic connectivity. There's all kinds of different protein markers in the brain that suggest that for these, the more you use your muscles, let's say, the better the function becomes.
Hey, you were talking about the time and the clock, and I get to think about something that I think is very strange, and maybe it's very common among us, but I can set my head on like an alarm clock and tell myself that I want to wake up next morning at 6 o'clock, 7 o'clock, 8 o'clock, and I always do that, maybe 1 minute before, but never later. And if I just sleep and have my alarm clock on, then I can wake up and feel that the clock is 4, but it's almost 2. So,
yeah, that's interesting. I experience the same. I've been waking up slightly earlier than normal lately, and now it only takes a few days, right? And now I happen to wake up before my alarm. So it's kind of hard to say how much your attention and your mind is really focused on. In the middle of the night, you have these every 45 minutes or so as we drift in and out of different stages of sleep. Maybe you become slightly mentally aware, like, okay, and this morning I'm going to wake up early. And even if that thought passes really quickly and you're not aware of it, those things might still be in your sort of attention in your mind as you're sleeping overnight and you manage to wake up slightly early. But that being said, there's also so many different rhythms in the body that are going on all at the same time. So I think it would be a mistake to say that it's just a mental function that allows you to wake up early like that your body can be programmed in a sense from all of your different rhythms of what time of day you receive sunlight, when you receive physical activity, and those can have an impact also.
You mentioned that the brain activity during REM sleep is quite similar to the one during awake hours. Is that also similar for like deep sleep, or is it just unique to REM sleep?
So we've mostly looked at different neural trajectories and even grid cell signaling during REM sleep. And in those states, we see it more often resembles waking, but the animal is sort of moving in and out of non-REM and non-REM sleep throughout the night. And the non-REM epochs, we haven't really looked at those very carefully. So it's possible that there's something going on there, but it's a very different state in the brain overall, so it's hard to say for sure.
Maybe we should just say, so everyone is on the same page, that REM sleep is in humans the kind of sleep that takes place when we dream sleep.
Yeah.
Yeah, and the others is when we, at least a different kind of dream, or no dream.
Yeah.
Hi. Do our brains have a limit or a capacity on how many memories we can have, or is it like limitless? Thank you.
You want to take that?
Yeah, that's a great question. It's a question, of course, that we would love to know the answer. But we don't have one. There are some collaborations that we have going on with both the math department and with AI, trying to understand like what might be the capacity in theory. But we certainly don't know. There's an enormous number of neurons in the hippocampus. So as Edvard mentioned, the number of pattern combinations that you can have if all of those cells are changing their activity patterns in new places, it undoubtedly is enormous, but I don't think there is a good estimate.
There was once something that's called a neuromyth, and that is that you only use 10% of your brain, something like that. Many people believe that, and it was said over and over and over again for many years, but it's complete nonsense. So it's neuromyth number one, because I mean, there is no limit to how much the capacity is very close to endless, and the more you use your brain, the bigger it gets. So, yeah. And you're always using all of your brain anyway. There's activity almost whatsoever, almost in all kinds of activity, with one exception. If animals hibernate, that is one of our postdocs is sitting there. He's actually doing that. So then the whole cortex turns off, apparently, which is quite amazing, because still these animals retain memories. So one of the puzzles. But other than that, the activity in the brain goes on all the time, everywhere.
We have time for one final question. And then.
Do you have the secret of how to make time pass by a little bit slower?
Do you have the answer?
I think we have some ideas, but probably not in the sense that you mean. I think, of course, as we get older, I experience this as watching my children grow up. Time seems to move faster and faster and faster. And maybe this is actually because as we're getting older, more things or more events are actually happening in our lives. So we have the perception that time is just flying by. For actually slowing it down, then any recommendation that would be related to that would be very dull, and to say then don't do very much at all, but that's certainly not a life worth living either.
Okay. Thank you, Christy and
Yes.
Edvard, and I will come up.
I don't know if I have some... I had one last question, which is actually not about neuroscience, but more about the interdisciplinarity, because I've also been working a lot with people from other disciplines. So I'm wondering, how do you cope with it? I was quite happy that you had some references as a philosopher. I'm happy that you referred to both Kant and William James. But actually, when dealing with this on a daily basis, how do you do it? I mean, because very often I think that we need to translate somehow in order to understand, because we do have different disciplinary languages. Yeah. There is no simple response to this, but yeah, it would be nice to just have your opinion.
It's not an easy thing, but I think bringing together people, have them to start speaking the same language, which is also a big step, right? Because initially, for example, when I met mathematicians and the topologists, they didn't understand anything. But in the end, it started to make sense. But then probably other way around you, we use words that not necessarily have to be so complicated. But I think working as a group of people who interact across disciplines, working on the same question, each one having a little to contribute, that helps. And then we try to develop a culture where everyone chimes in with their background. I know.
Yeah, I think that's a fantastic answer. For me, I think that the environment that's cultivated in the lab is a really fantastic one for very open communication. So we have people with different backgrounds scientifically coming in the lab, and we give presentations to one another, and I don't think anyone is afraid to jump in and say, well, this doesn't make sense to me, I would have thought about it this way. And also in the process of publishing articles, that we're trying to send the manuscript to an array of people who might read the words that you are writing and just have a completely different and surprising take on what it is that you're saying, and maybe alerting you to the fact that not in all fields would what you're saying be perceived the way that you think. And so I think just constantly communicating your science with both your lab mates and the broader scientific field is really the best solution. Just hearing what other people have to say about the questions you're asking on is probably most important.
Thank you, Christy. And thank you, Edvard.
Okay.
Thank you very much. I'll plan to say this in Norwegian, but now we have been talking English all the time, so I do it in Norwegian anyway. Så tusen takk til alle som har møtt opp. Jeg tror det her har vært veldig givende for veldig mange gode spørsmål, og very nice talks. So you open a field, and you even said something about how it is to work in the laboratory, language-wise, in a way. Så, takk til innlederne, takk til Asta. Og jeg tenkte jeg skulle veldig kort si at vi skal også komme tilbake hit til Litteraturhuset 13. oktober. Da har vi et møte som handler om fordeling og verdiskaping. Og da kommer Kalle Moene, professor i samfunnsøkonomi, Guro Angel Gimse fra NHO, Roar Aas fra LO, og vi skal også da ha en panelsamtale ledet av professor Ragnar Torvik, som også sitter i styret i DKNVS. Og med det så håper vi at mange finner veien tilbake, og tusen takk til alle for godt oppmøte og gode spørsmål og diskusjoner. Tusen takk.